Okay, so we start this new session of the Young Seminars. We have two speakers, as usual, Serena Di Santo and Gianmarco Spera. We start with Serena, which I briefly introduced. She studied in Parma University. Then she had her PhD in a joint project between Parma and Granada universities, with under the supervision of Raffaella Burioni and Miguel Angel Muñoz. Then she had two postdocs, one at SISSA with Alessandro Treves, and the second at Columbia University, New York, with Ken Miller. Now she has a tenure track position in Granada University. The title of the talk will be Quasi-Universal Scaling in Mouse Brain Neuronal Activity Stems for Edge-of-Instability Critical Dynamics. So you can start, Serena, please.
Thank you very much, Federico. I'm gonna share. Okay, so talking about scaling variance in neuronal system and how it relates to criticality. First of all, I'd like to thank Guillermo, who is the main author of this work. He recently got his PhD, looking for postdocs, just a little shout out, and Miguel Angel Muñoz from University of Granada. This is the brain of a larval zebrafish. So this little fish with a transparent skull. And here you see a movie of its neuronal activity recorded at the single neuron scale. So here you can see actual single neurons. And of course, one can say that this system is in a state of perpetual or ongoing activity. And you can observe also that within this ongoing activity, there are localized activation events, such as like these small ones that involve few neurons that coexist with delocalized ones, the ones that involve up to the whole system. And of course, the hypothesis for a statistical physicist that is very tempting is to say that the brain might work near a critical point, so at the edge of a phase transition. Okay, so first question that we're going to ask is, does neural activity show genuine scaling variance? Let me notice that, well, okay, yeah, let's start with this. Does it show genuine scaling variance? And for the last 20 years or so, people have been asking this question and mainly addressing it by analyzing neuronal avalanches, which are these cascades of activity, cascades of events interspersed with periods of silence. And the claim is that if one measures the statistics of the sizes and durations of these events, one finds power laws that are just a signature of scaling variance with some degree of finite size scaling. However, this measure is not very robust. It has been criticized a lot. For example, it is dependent on the binning, so the discretization that one does of these continuous signals, as well as sensitive to subsampling. In fact, avalanches become smaller and smaller the more the system is subsampled. No? If I measure less neurons, my avalanche will be smaller. So we would like a better tool to test the scaling variance. And of course, the renormalization group is the go-to if one wants to do things in an elegant way. And here, just to set the language, let's say, the renormalization group consists in either iteratively grouping together neighboring units. In this case, of course, is Ising 2D. And when the system is self-similar, then the statistics of the system stay the same at every scale. They only depend on the cluster size, let's say, on the RG steps that one is taking. And here in this visualization that is, I think, very beautiful, one can see that how this zooming out process kind of is able to reveal genuine self-similarity as well as deviations from it. Okay, so the idea is, can we do something similar in the brain? Basically, in this system, we have a big problem. That is that we usually, most of the times, do not have information about who is neighbor of whom, or more precisely, we don't have information on who is neighbor of whom together with information of activity, on which neuron is active. And so to solve this problem, Lino and Meshulam, in the group of Bill Bialek, proposed this phenomenological RG scheme, which consists in, instead of blocking together neighboring units, blocking together highly correlated neurons. So in this case, we don't need the information of the JIJ, the underlying network. So what they did was the following. They took their activity, in this case, yeah, this kind of opto physiology, these optic recordings, whatever it is. So they extract the activity for a single neuron. They calculate the covariance matrix among neurons. They pick the most correlated couple and they group them together. So they form a super neuron, or like a cluster of size 2. And then they keep doing this with the next most correlated unit, the next most correlated couple. They create a new unit, etc. This is step 1 of RG, and then they keep doing this iteratively. So this is the scheme that they propose. And then they show that with this scheme, they can reproduce some sort of scaling variance that I'm going to show now for this work that we did, taking this data set from Steinmetz et al. At the University College London. They recorded from different regions of the mouse brain, both superficial and deep regions. And the mouse in this case is at rest. So what we do is to choose a time resolution for each region. And basically in this case, this is the geometric mean of the ISI distribution, the ISI, which is interspike interval distribution of the neurons of this region. And then we coarse grain by using this RG scheme that I just outlined. And then following Meshulam, we search for scaling variance, namely, or like more specifically, what we analyze is the covariance, the variance across clusters. And we see as a function, let's say, as a function of cluster size. So the variance across clusters scales, as one can see here, with non-trivial exponents, that would be these dotted lines here, with cluster size. The same is true for probability of finding a silent cluster, and the same is true to a certain degree for characteristic correlation time. In this case, each color is a different brain region, as one can see here. So what one can observe is that not only there is a quite a good scaling across cluster sizes for a specific region, so within a specific region, which is indicative of the fact that the physics is the same at every scale, but also across different regions. So in this case, and in this case particularly, and for correlation time, it's a little bit more dispersed, but still, this is a biology experiment, and it's not trivial to find clean stuff in biology. Another thing that one can look at is the covariance matrix spectrum within clusters. And then what one can see is that for different cluster sizes, here different colors is different cluster sizes, these spectra collapse very nicely on top of each other. This is for one specific region. And this is, again, another sign of the fact that there is genuine scaling variance in the system. So I don't know if I convinced you, but at least I did convince myself that in this neuronal system, one can actually observe scaling variance. And then the next question is this scaling variance related to criticality? The underlying idea, as I said at the beginning, is that criticality not only could explain the scaling variance, but could also, like from a different kind of part of the literature, could also bring about some functional benefits, such as, let's say, optimal, say, maximum computation capability, sensibility to stimuli, etc. More specifically, what we ask is, can we quantify how close an empirical system is to the edge of a phase transition? And we take the simplest possible model to answer this question. This is a linear rate model. The variable is x, I is neuron I goes from 1 to n, and x is a continuous variable describing the firing rate of the neuron. And JJ is this connectivity matrix, and then one has this external input, let's say, in the form of a noise term, which is uncorrelated white noise. And of course, the brain is not a linear model, and it's much more complicated than this. But still it's still quite a mystery. However, this model is quite remarkably good as an effective model, as a linear response approximation for more complex systems. So basically, the results that one can find with this model generalize well to nonlinear models, let's say nonlinear fastness functions here, nonlinear models, models of spiking neurons. So instead of continuous, it would be discrete variables, and neurons, systems with two populations, excitatory inhibitory, etc. So back to this model, basically what one can say is that this model becomes unstable when the maximum eigenvalue of the connectivity matrix becomes larger than 1, because of this -1 here. And then if one could measure this JJ in data, so if one could measure this connectivity matrix in the data, in principle, one could already state if, according to this linearized model, the system is close or not to the edge of instability. And here I want to show that it's not that far-fetched to think that one can actually measure this in actual experiments. People measure if neurons are connected, which neurons connected to which, and how strong is their connection. But of course, to measure the eigenvalues of a matrix, one needs the full matrix. Cannot have a subsampled matrix. However, people also do this. Experimentalists also do this. So here you have the full connectome of a fruit fly. Anyway, it is very challenging to do these measures, as you can imagine, experimentally, and therefore, let's say we're going to make a little bit more effort from the theoretical side. So we're going to choose this Gij to be a random matrix with Gaussian entries with 0 mean and variance g²/n, and here basically I'm plotting just different matrices, and then for this matrices, a known result from random matrix theory says that the spectrum of these matrices will be uniformly distributed within a disk of radius g. So the radius of the spectral radius of the matrix will be related to the variance of the entries. Cool. So if it is difficult to measure the connectivity matrix, it is much easier to measure experimentally the simultaneous activity of many neurons. Nowadays, up to hundreds of thousands of neurons, tens of thousands or hundreds of thousands of neurons. And next, what I'm going to do next is to show how an estimate of lambda max, of this maximum eigenvalue of the connectivity matrix, can be obtained from a measure of the pairwise correlation across neurons. So we use these recordings from the activity of neurons to estimate the stability of this linear rate model. Okay, and I'm going to present two methods, one from David Dam and colleagues in the group of Moritz Elias, and one from Yuhu in the group of Hans and Poliski. For both methods, what one does is to define this long-time window covariance matrix. So basically, first of all, one defines a long-time window, this delta t, and integrates the activity of this whole time window. And then one measures the covariance across this time integrated signals. And this is what we call long-time window covariance matrix. And this matrix has a simple relationship, as you can quite easily see, with the connectivity matrix, J. By using this relationship and some other more complex, say, framework from the physics of disorder system, what Damen and colleagues were able to do was to find this very elegant relationship that connects the maximum eigenvalue of the connectivity matrix with the relative dispersion of the entries of the covariance matrix. So again, if we go back to this model, what this relationship is saying, what this formula is saying is that the variance of the entrance of the connectivity matrix is related to the variance of the covariance matrix. And this relationship turns out to be, I mean, these points are simulations for 1,000 neurons. So this formula depends on n, which is the number of neurons, but it is quite well, say, So the simulation is quite consistent with the theory for this simulation of the linear rate model. And what I wanted to say is that 1,000 is a number of neurons that is pretty much the same as what we have in the experiments for each region. But remarkably, what is interesting here is that this measure is insensitive to subsampling, right? Because this is just considering the statistics of the covariance matrix, relatively insensitive. And moreover, this so for our peace of mind, Dama and colleagues have shown that this formula is actually quite robust to changes in the dynamics. For example, going to networks of excitatory inhibitory neurons, or even networks of leaky integrated and firing neurons, or spiking neurons, very nonlinear. Okay, so we take this method and we just apply it to the same dataset as before. First of all, we need to define this long time window. In this case, what we choose is a time window that is longer than the autocorrelation time. We compute this spike count or long time window covariances and the delta, which leads us to an estimate of lambda max. In this case, we call it G hat T, so empirical estimate, which is the empirical estimate of the distance to criticality, right? And here's just a little technical note that is every different region has a different number of neurons here. So if we want to compare across region, we want to extrapolate to a common number of neurons, 10 to the 4th in this case. And then we can show for each region how close it is to the edge of instability, so G hat D equal to 1. All we can see is that all the considered regions are pretty close to this edge of instability. And next, I'm gonna consider this method 2, which is basically a different way of estimating this lambda max. And this was proposed by Yuhu and Sompolinsky very recently. And what they do is to actually compute the full expression of the spectrum of the covariance matrix, the eigenvalue spectrum of the covariance matrix. In particular here, this is still for a linear rate model with the random Gaussian connectivity. And in particular, what one can see is that for G goes to 1, so G approaching 1, which is again the edge of instability, the support of this distribution goes to infinity, so becomes larger and larger. And in particular, this expression becomes a power law with exponent 5/3 or minus 5/3. Again, for our peace of mind, they could show that for a nonlinear system, so now we have this transfer function stage. This formula still holds. However, this is just basically for a linearization of the system, which is basically considering let's say a bias in the estimate of G. But this doesn't matter because for our purposes, let's say we just consider the fact that the linearized system is close to instability or not. So let's say it works also for nonlinear systems. So we do the same as before. We take long-time windows, compute covariance matrices, and then what one can do is to fit the empirical distribution of the eigenvalues of the covariance matrix with this formula, right? Where G is the free parameter. And then one can actually infer this value of G. And in this case, this is for one region, one finds this value that is quite close to 1 again. And in particular here for comparison, we plot this green line, which is the Marchenko-Pastor distribution, which is what one would expect if the units were independent. Okay, and of course this distribution fails to capture the detail of the empirical distribution. So that was for one region, but one can compare this measure of G hat S versus G hat D with G hat D, which is the two different estimates of this distance to the edge of instability. And the two are in good agreement with each other and indicate that the system is, let's say, to a larger or lesser extent, every region is to a larger or lesser extent close to the edge of instability according to both measures. So basically, is this scale invariance related to criticality? We can answer to this question, and the hint here is yes, at least like within the limits of this model. Summary: to conclude, I guess, strong signatures of quasi-universal scale invariance are observed in resting state neuron activity across brain regions and experiments. Here I didn't show that we analyzed different experiments, physiology, but also electrophysiology, so different kinds of experiments. We found evidence that this scaling may stem from the edge-of-instability critical dynamics. In that basically we exploited this relationship between lambda max, maximum eigenvalue of the connectivity matrix with the say different, either the statistics of the long time window covariance matrix or the spectrum of the same matrix. And both measures indicate close to critical behavior, no? That these systems are close to the edge of instability. So bottom line is that operating near the edge of a transition seems to be an excellent solution that nature found to perform robust computation collectively. So again, evolution seems to have found this idea and exploited it. And next, just 1 minute maybe on the perspective. So what's next? Basically now, Ruben and Carles, who are two PhDs in the lab of Miguel Angel, are investigating the multi-band aspects of this problem, basically going beyond this idea of the long-time window covariance matrix, because one is integrating over a long-time window and therefore kind of averaging out all time-dependent effects. However, we know that the brain, in the brain is very relevant, that there are these kind of transient synchronization phenomena, oscillations and traveling waves and all sorts of interesting time-dependent phenomena. So basically the idea here is to define, instead of a long-time window covariance matrix, a covariance matrix as a function of frequency. So what is also called well, we call it a frequency-dependent covariance matrix, but it's also called a coherence matrix. And then basically study how close the system is to criticality at a specific frequency. Again, thanks to Guillermo and Miguel Angel. This is the paper where you can find more information, and thanks to you all for your attention.
Okay, thank you, Serena. Is there any question from the audience? Well, meanwhile, maybe I have, oh yes, there is one. I see one hand raised. I don't know who is him.
It's me. Hi, Serena. Relating to the last part of your talk, how do you define this time window? I mean, do you have an experimental estimate, some kind of reasonable definition of the time window that you have to use? To get a reasonable result for the lambda max in experimental cases, not in.
Yeah, can you still see the screen?
Yes.
Yes, okay. So basically, in the experimental case, in this data set, what you want to do is, on one hand, you want to make to have a long time window, now to be able to, to apply this formula, because delta t in the formula goes to infinity. So it needs to be long enough. But on the other hand, maybe actually I forgot to mention it, but you need to average over long time windows. So you don't want it too long, because you still want to divide your whole data set into chunks so that you can average over these samples or different realizations. So basically what we do is to take a trade-off, and as long as possible, but still that allows to divide it in many chunks. In this case, at least it should be longer than the autocorrelation decay time. And this is what we're showing here. No, we measure this autocorrelation time for region, like these different lines are different neurons. This is the average. And then we take a window that is the size of 3 seconds, which is at least autocorrelation decay.
Okay. Thank you, Serena.
Any other question? Maybe I have not a question, just a curiosity. You mentioned at the beginning that there is, of course, some upper cutoff in this power law behavior. So this upper cutoff can be due to the finite size, of the brain, but also by the fact that this maximum eigenvalue is not exactly 1, so that you are close to the critical point, let's say, but not exactly at. So
Yeah,
is there any study which is trying to relate the maximal eigenvalue with the place where you see this cutoff to see if they are connected?
So let me see if I understand. Maximal eigenvalue of the covariance matrix, right?
Yeah.
Yeah. Okay. So the idea is, yeah, basically, let's say one observation towards that. Well, of course, to estimate this tail, one always has these problems of sampling, right? Because okay, it's true that this is quite insensitive to subsampling, but still, I mean, this covariance matrix will have the size of your, let's say, number of neurons by number of neurons, which is not infinite, right? And then you have to make a distribution. So you will have some kind of, it's not going to be easy to find.
Yeah, I can imagine.
Say to find a good fit. One interesting thing to that, say in this sense, is that basically, is this true? No, I was saying something different. I was saying something about the spectrum. I wanted to say something about the spectrum of this matrix, of the connectivity matrix. About the maximum eigenvalue of this one, which would be this lambda plus. Yeah, I don't know of anything specific that goes in that direction. I think because of this kind of sampling problems.
Yeah. Okay, you understand what I meant. I mean, suppose you are, for instance, not in a percolation problem, you're not exactly at percolation, but the probability is slightly below, then you will not have clusters of all sizes, only up to some maximal length. So I can imagine that something similar could happen here. So, and maybe there is a way to relate the distance from criticality to the upper cutoff, but of course I understand that experimentally it could be very hard to see that.
Yeah, as in related to the finite size of the system.
Not only related to the finite lambda, which lambda is not exactly 1. So you are not exactly at criticality. Okay. Any other question? I don't see any. So if this is the case, we can move. Thank you, Serena, again.